Hello, and thank you so much for joining us here today for, I think, the sixth or seventh in our series of webinars from Triverity Laboratories. I'm Wendi Young, I'm the CEO at Triverity. Today, we're going to be talking about analytical method validation. As she just said, the webinar is being recorded, and we will provide this out. We'll provide a link on LinkedIn and via email as soon as the recording is available. If you have any questions, you can put it in the QA. This one, I think, is going to go a little bit long. So so we might not have time for questions, but we'll make sure that we get answers to you for any questions that come up. A little bit about me, if this is your first webinar. My background is in analytical development. I've spent almost 30 years now doing method development and have either developed or led a team that has developed thousands of analytical methods. So method validation is my jam, and you'll probably get a sense of that as we go through. There's a lot to talk about here. So let's just get started. The applicable regulations for method validation, 21 CFR Part 211, 165, Section 3 states that the accuracy, sensitivity, specificity, and reproducibility of test methods employed by the firm shall be established and documented. Such validation and documentation may be accomplished in accordance with 211-194, which I have below, and that is the laboratory record section. So there's also USP for method validation, USP General Chapter for verification of compendial procedures. There's the lifecycle management and then transfer of analytical procedures. We're going to talk about all of those, but mostly we're talking about ICH Q2AR2, which is validation of analytical procedures. The forthcoming webinar will cover ICH Q14, which is analytical procedure development. We might do that with Q12, those two together, but we'll just touch on analytical development or procedure development here. So what is method validation? Method validation is showing that the method that you're using is fit for the intended use. It is suitable for use. It is required for release testing. It's required for stability testing, for drug substances, drug products, also things like excipients, critical excipients like preservatives or antioxidants. And then some performance methods like dissolution. And method validation should be phase appropriate. And we're not going to talk a lot about phase appropriate method validation because really if you're going into a phase 3 clinical trial, your method needs to be validated. If it's before phase 3, it needs to be qualified. And qualification is just, you need to have an assurance. I like to say that your method is validatable. You know that if you go into validation, it's very likely that it's going to meet the validation criteria. So you want to have done enough method development and method assessment to make sure that the method's going to be work that it's suitable, but you don't actually have to draft a protocol and write a validation report and do all of those things. So where the method validation fits kind of in the QBD, you identify your CQA decision points. So are you testing at release? Probably you're testing at stability. Do you have any in-process controls? Where are you going to be utilizing this method? Then you need to define what your analytes are, what your matrix is, and what the range of the method has to be. We have had a number of situations where we've had people come to us and say, we need to get a method validated for this. We're like, okay, what's your analyte? They haven't even gotten to the point where they know where they're testing, what their specification is, or even what they're testing for. So a lot of this work has to happen in the development before you even get into method validation. You need to be thinking about what do we need to test? What is our specification range going to be? Because then you can look at it and say, okay, well, I know based on the specification what the allowable error in my method can be. If you're looking at an API method, an assay method for an API, and your specification range is 99 to 101, or 98 to 102, your method error better be pretty tight, right? So then you take your all of that and you translate it to your analytical target profile. What is the method? What is the criteria that the method has to meet, right? Then with that, then we start with product understanding. Then we figure out what our CQAs are. We come up with our control strategy. What are we testing? When are we testing? We determine our analytical target profile. What does the method have to be? And from there, then we can develop the method, do our risk assessments, move into validation and routine use, and then we go into lifestyle management. And that's where we are looking at our system suitability, any changes that we need to make to the method, reviewing revalidation, all of that kind of stuff. This table or this figure is directly from ICHQ2, and it indicates kind of where or how ICHQ14 and Q2 go together. So Q14 is what are the objectives of the method? What is our ATP? What information do we have? From there, then we can draft our validation profile or protocol, our validation strategy. Um, and then that goes into executing the validation. That's at the bottom of the figure, evaluating the data, drafting our report, and then going back into Q14 with the lifecycle management. So with the lifecycle management, if you make a change to your method after it's been validated, and we're going to hit on these three really quick before we get into kind of the nitty gritty of method validation and a lot of the details. So lifecycle management, if you're validating a method and you make a change, you decide that maybe you need to add a sonication step, maybe you're not getting full extraction, you've seen some variability. You need to look at the method validation that you did previously. What did that change? What part of the performance characteristics would be impacted by that change? Likely it's accuracy. If you're thinking you're not getting full extraction, maybe accuracy has been impacted. Very likely precision if you've been having variability issues. So then you would look at your method validation. We don't need to repeat linearity. We probably don't need to repeat specificity, right? So then you say, okay, what part of the validation do I need to repeat? When you do a partial revalidation, you need to make sure that you provide a justification of like, this change didn't impact these performance characteristics. We don't need to look at that. Method transfers are when you're taking a method from your lab and transferring it over to another lab. An example of that is a analytical development lab transferring to a QC lab. It might be based on your knowledge of the receiving lab. Maybe the receiving lab has the same instrumentation. Maybe it's your QC lab. You really have a good understanding of their capability, their knowledge. Maybe they have prior experience. Really, you just need to do a comparative analysis of a sample. You know, the analytical development lab tests the sample 6 times, the receiving lab, the QC lab tests it 6 times, and you compare it. Maybe you're sending it to a completely different lab and you're not sure it's going on, you know, your adjuvant, their waters. You need to check specificity. Maybe you need to check linearity because there might be some response issues. Maybe you're using response factors. Maybe you need to check LOQ. Or you're in a situation where the method has been validated, maybe the lab's shut down, there's no way to do a comparison. Maybe you just have to completely revalidate that method with the transfer. So you really have to take a look at your situation, critically look at what do we need to do to ensure the success of this method, one, and then two, to support the validity of the data that's going to be generated with it. And then verification is verification of compendial methods. If there's a compendial method, you don't have to do a full validation. You just need to validate the performance characteristics that you think are critical to that method. If it is let's say it's an impurity method, definitely specificity. You want to make sure that your system is going to be specific for all of those impurities. If it's also impurity method, you want to test the sensitivity, the LOQ of the method. Make sure that you're able to see down as low as you need to, that you can get the reporting limit, all of those kinds of things. Some of the methods need to be stability indicating, and this is when the method is going to be used to generate stability data. So stability indicating method is a validated quantitative method that can detect changes in relative quality attributes of product during storage. That's the ICH definition. So what that means is you need to have a method that can detect changes in the product or the API over storage, whether that's in a chamber or wherever. If you have degradants that could be colluting with your API peak, you might not be able to detect change in your product. You're not going to see that degradant growing. So you need to do some work to prove that your method is stability indicating. So samples containing degradation products should be included in the validation study. In that bullet it says containing relevant degradation products, and I think that's kind of important. When we do forced degradation, we're really hitting that with a lot of like high acid content, oxidative, really high temperatures, really trying to force that degradation to show that we've got specificity of the method. Not all of those degradants are going to show up in the real world. So when you're looking at method specificity with forced degradation, you really need to take a critical eye and say which one of these might actually show up. And that's where you looking at stress samples. So that would be accelerated storage or maybe a little bit higher than accelerated storage, but not bombarding it with really high pH or low pH, lots of UV radiation, right? So really making sure that that you're not going overboard with the forced degradation and the specificity there. System suitability is the set of standards that, and this is really focused on HPLC GC chromatography, but it's a set of standards that we run at the start of a sequence to show that the system is suitable for analysis at that time. It typically is some blanks, maybe an LOQ, replicate standards. And then we also run maybe an impurity composite, and you're looking at resolution of some critical pairs. Um, it, your system suitability should be established during drug development, or sorry, during method development. You need to figure out what criteria the system needs to meet to make sure that the data is valid. You need to do this prior to method validation. And some of the critical metrics that we're looking at is RSD of those replicate injections of a standard, signal to noise of your LOQ, maybe tailing, sometimes plates, resolution of critical pairs. And then we want to bracket our samples with a confirming or a check standard to make sure that the method stays suitable. System suitability should be established during method validation. If you're not meeting your system suitability during method validation, that's an indication that your method or your system isn't robust, that there's something wrong with your method. It could be that you find out that maybe you hadn't equilibrated, but you need to do an investigation and look at that system suitability failure and figure out what happened. So your analytical procedure validation strategy is what your plan is to validate. It's developed based on your knowledge of your procedure and the intended purpose of the method. And it is supposed to be designed to demonstrate that fit for purpose. So you want to set your validation strategy up to meet the analytical procedure objectives. And those are your performance characteristics. And this all comes into, we're going to get into this in the Q14, but it comes into the performance characteristics like linearity, accuracy, precision. And then the performance criteria, which is the acceptance criteria associated with those performance characteristics. Recovery of 95 to 105%. Or an RSD of less than 10%, something like that. Those are your performance characteristics and criteria. We're doing method validation. We need to start with method development. Then we go into late stage development, which is the forced deg and the robustness. Then we determine, based on all the data that we've generated and understanding of our specifications and the intended use, our validation strategy. We draft our protocol, we execute the protocol, and then we draft a report. And then comes some life cycle. I would say life cycle management. Our validation protocol needs to have at the beginning information about the intended purpose of the analytical procedure. So this is an assay method for API X in product Y. If there is or if there's different formulations, it's really important that you get you're really specific about what the formulations are, what this method is being validated for, what this method can be used for. Because when we do method validation for drug GMP, we're validating for a specific formulation, a specific formula, a specific strength. It can be that we're validating for multiple at the same time, but we need to validate for each one of those. If a new formulation or a new strength is added, we need to evaluate the method validation and make sure that it is either under the scope of that method validation, or we need to do a bridging study to expand the scope of that method to include that product. Your validation protocol needs to include the performance characteristics, your performance criteria, what the acceptance criteria is, any justification for reduced validation. Oftentimes we use the same chromatography for our API as we do for our drug product. And in those cases, we don't need to repeat linearity again if it's the same concentration, if it's the same range, same chromatography. So in our drug product validation protocol, we'll reference our drug substance validation and state that the method parameters are the same. And then in our validation report, we'll actually re-report all of that, because we like to have a validation report that is comprehensive. You don't want to have to provide a report to a regulatory body for your drug product and then also for your drug substance. If you didn't need to, you probably would anyway, but it's better to have all of the information in the same place. And then we want to have detailed instructions for the execution of the validation. And this is an example of one of our validation protocols over there, where we've got method precision, and we say method precision will be demonstrated with the analysis of 6 samples per draft 116, T116, Appendix 1, Section 8. So we're very specifically saying you're going to test 6 samples per the draft method, which we attached as an appendix, and test it per Section 8, which is our sample preparation procedure. We go on to say how to calculate it, what exactly you need to calculate. We tell you to report a representative chromatogram in the validation report, and then we give acceptance criteria. So we're very, very specific. If this was an accuracy section, we would be telling you how to make the spiking solutions. We would be telling you how to spike the samples. You want to be very specific so that the validation protocol can be executed easily. And then your validation protocol should take into account the number of replicates that are going to be used in routine analysis to generate your reportable results. So if you are taking 3 replicates and averaging that for your reportable results to compare to your specification, that needs to be taken into account in your method validation. It's possible that you're looking at some product variability. Maybe the capability of your manufacturing process isn't that great, and so you've got tablets. So you're doing a composite. You need to take that compositing into account when you're doing your method validation protocol and your validation study design. Your validation report needs to be a summary of your validation strategy. Any justification of reduced validation needs to be indicated. You need to have data summaries. I've got an accuracy table there that's massive. You want to compare your results to your protocol acceptance criteria. For linearity, provide the plots of the data. So in this case, we had, I think it was, I don't know how many that is, maybe 10 different analytes. And so we have a plot for each one of those. You want to also provide representative chromatograms for each data set. You don't need to provide, if you're doing an N of 6 for method precision, you don't need to provide 6 chromatograms, just provide the first one. We're very consistent in always providing the first one so that it doesn't look like we're cherry picking the best chromatogram to put into the report. You also want to list any deviations that you had in the execution of the protocol. Example of a deviation would be method validation did last week. I screwed up the linearity preparation. I had one of the volumes wrong, and so we had to write a deviation to correct that, which, that's not a big deal. If you have a failure in your acceptance criteria, that's a really hard deviation to write. It's hard to write a justification to widen acceptance criteria. So you really want to think about it. What really is your failure? What really is no longer acceptable when you're writing it? What is your method capable of? You want to have data references so you can go back to the raw data. And then a validation conclusion. We do conclusions for each parameter, each section. So a linearity conclusion saying that the method is linear from this range to that, or this from X to Y. And then we also do a final validation conclusion that says the method is essentially fit for its intended use. ICH and USP classify analytical procedures, and the classification helps to identify what parameters need to be evaluated for method validation. So they're broken down into identity, so identification tests, impurity, which is quantitative, impurity, which is a limit test, where we're not generating a number, we're just saying it is either above or below this certain value, and assay method, performance method like dissolution, and then multivariate methods, and we're going to talk about that here. So a multivariate method is where you are using a model to assess the quality. To me, I look at this kind of as like a secondary analysis. So an example is I worked for a testing lab where I generated a near infrared model for silage. So I took moisture, fat, and fiber data and analyzed the NIR spectra of the silage and inputted the data that we generated for, I think it was probably 500 different lots of silage to create this model so that we could test subsequent lots of silage using that model and not have to actually do moisture, fat, and fiber. We could just analyze it by NIR. So that's a multivariate analytical procedure. So there's 2 phases. One, you need to do your model development, and you need to make sure that the data that you're putting into that model is anchored to a qualified reference method. So you want to use a like the USP moisture or water content method or those kind of things. You want to have it be a qualified method the data that's going into this model. And then you want to validate using challenge samples. So samples that aren't, that have different analytes in it so that you can make sure that your model actually works for the analytes that you're testing for. The different validation performance characteristics are range. So what is the reportable range of the method? What is the working range of the method? Specificity or selectivity, linearity, accuracy, precision, limits like LOQ and LOD, limit of quantitation, limit of detection, robustness, which is how the method responds to deliberate small changes, and then solution stability. Here's a table from ICHQ4. It's a little bit different than the USP, and I'll tell you where in just a minute. But it lists in the first column, the performance characteristics, some of which we just talked about. And then across the top in the columns, it has identity, impurity, whether it's quantitative or limit test, and then assay. And these correlate to USP categories 1 through 4. So for identity, if it's got a plus, that's a characteristic that you need to evaluate in your method validation. Validating identification tests are the easiest. It almost seems like it's not worth the paper for the protocol because it's so straightforward. But you're really looking at specificity. Can I discriminate the analyte that I'm looking for against other similar analytes, right? When we're at impurity, so the quantitative impurity, we're looking at specificity range, which is our linearity, lower range limit, QL, which is LOQ, but no LOD. We'll talk about that in a minute. Accuracy, and then precision. Precision is repeatability, and then also intermediate precision. And that 5 there states that if you're doing reproducibility, which is like a method transfer to another lab, you don't necessarily need to do intermediate precision within your own lab. And that's true for quantitative impurities and also the assay. If you're doing a limit test, is it there or is it not there? Specificity and detection limit. How low can I see? What is my detection limit? And then can I discriminate that analyte from other analytes? And then for assay, we're looking at everything except for looking at the lower range because we don't have to look at really low levels, so the LOQ and the LOD don't really matter. Reportable range should be derived from the specification. This is the range in which you need to be able to report a result. And you have to confirm that by demonstrating at response, which is sensitivity. Can I see that low? Can I see it at low concentrations? Accuracy and precision. So the table to the left there is a list of different analytical procedure types, like the classification of the procedures that we just talked about, and then the reportable range. So the easiest assay, what it's stating here is that you need to have a range that is at least 80% to 120% of your target concentration or of the specification acceptance criteria. So if you are 90 to 110% for your drug product, then you need to be 80 to 120% of that. Typically, we would go just 80 to 120 or 70 to 130% of the target concentration for a product. Potency is for like biologic methods. Again, that's plus or minus and then plus 20% of the acceptance criteria. Content uniformity is 70 to 130 of the content. Dissolution, this is about the only time we're going to talk about dissolution because it truly is its own beast. So I don't want to get into this because it would take up, we're already probably going to go late. If you're interested in dissolution, I highly recommend going to the Agilent website. There is, Agilent has an expert in dissolution, Ken Boda. He's got a series of dissolution. There's probably 30 of them, but there's like a core dissolution on method development, method validation, if you are interested, and then qualification of your disso apps and everything like that. Really good series. And then impurity methods, you want to look at from the reporting threshold. So according to ICHQ3A or B, what is your reporting threshold? How low do you have to see? Up to 120% of your specifications acceptance criteria. Now, I would say, I would take that 120 with a grain of salt. You can justify higher, you can have a wider range. When you are in product development, method development, you have limited knowledge. And if you have a degradant that you see growing, you set a specification based on where you think it's going to be. It could be different. Going back and revalidating a method it's really not something you want to do, especially for extending the range of an impurity or two, right? So if it's a degradant, I would probably go wider than 120% of the specification. because it doesn't really matter. You don't want to go crazy. You don't want to be like, if it's 0.05% to 10%, that's not reasonable. You're not going to have 10% of an impurity. You might have 0.5% of an impurity. So maybe it's 0.05 to 1%, right? So looking at that and being I like to be a little bit more conservative in my impurity ranges. And then purity as percent area. We don't see a lot of area percent methods. You want to go 80 of the lower specification limit. And then since it's an area percent, it's not going to get above 100, really. I mean, you'll have some with method variability. Well, you're not going to get above 100. So up to 100 or your specification limit, but we don't see a lot of area percent methods. We're going to walk through now all of those different performance characteristics, and then we're going to go into different methods, and I'm going to give you examples of kind of the different types of methods that we validate and how we would validate it. So specificity and selectivity. Specificity is demonstrated through absence of interference or by comparison to an orthogonal procedure. An orthogonal procedure is one where something is significantly changed. I like orthogonal procedures if we're talking HPLC to switch from methanol to ACN. We want to change the specificity of the method in some fashion, maybe putting on a different column. And what we're doing when we use an orthogonal procedure is looking to see the profile of your API and impurities. Do we see any new peaks that are showing up that were previously co-eluting that we didn't see before? And that really shows us if we run it by an orthogonal procedure and we don't see anything new, it's very likely that our method is specific. But typically we can show specificity. We've already run through a ton of method development to make sure that we have accounted for all of if we're doing an impurity method, we've accounted for all of the peaks. So hopefully by that point, you know what your impurities are. You can demonstrate it by the analysis of all the analytes, the matrix, the blank, showing that everything is resolved. Every analyte can be individually analyzed. You can use peak purity for HPLC DAD to show that there's only one analyte in the peak, and that is a assessment of the spectra of a peak to make sure that it doesn't look like there's two different fingerprints in there. It typically is reported as either, I think, like usually we see it as like 1,000, so 999 would be very pure. If you're getting below 990, we would say that maybe there's some interference in there. You also want to look at critical pair resolution. So critical pair would be the two peaks in your method that elute the closest to each other and what that resolution is. Selectivity is when the method is not specific. An example of this would be a assay method, maybe your dissolution chromatography, where you are running a really short method, and you might have some impurities that co-elute with your API, but the concentration is so low that those impurities are never going to register. Maybe they're at 0.1% and you're running a really low concentration. You're not going to see those impurities. So the interference is minimized and the method is still demonstrated to be fit for the intended purpose with the method validation, showing the accuracy, showing the precision. So for specificity or selectivity, where the impurities are available, so if they're commercially available or they can be created, you want to look at spiked samples or forced degradation stress samples with mass balance. And mass balance is when we look at the assay and the impurities combined together of a nominal sample and compare that to the assay and the impurities combined together of a forced degradation or stress sample. We want to compare those two and make sure that we're getting close to the same value. If we're not getting close to the same value and the stress sample is low, maybe it's about 5% lower than it was before, that indicates that there might be some impurities that aren't being detected by your method. Whether they can't be detected by UV or they are co-eluting with another peak, that mass balance helps us to determine if that's happening. If the impurities are not commercially available, you need to run samples with impurities. And commercially available means that you can reasonably purchase them. We can get almost anything synthesized, right? But how much is that going to cost? Typically, if you're purchasing an API, the API manufacturer provides the impurities to you, or you can go someplace like Sigma and purchase them. So if you're not able to purchase the impurities, maybe you don't have it identified yet, you want to look at samples with impurities and then compare that to an orthogonal procedure to make sure that you can get that specificity. For linearity, we want to see a minimum of 5 concentrations across the range of the method. I typically do 6 for impurities, lower level, and 5 for assay. You want to plot the response versus the concentration. I've got a poorly labeled graph there for you. So the concentration's on the bottom of the graph and the response is on the y-axis. You can mathematically transform the curve using like a log function if you need to, if it's not linear. If there is a nonlinear response, and some methods are nonlinear, then you don't have to evaluate like the correlation coefficient, but you do need to demonstrate the method's ability to accurately measure across the range of the method, which you have to do for accuracy anyway. You want to report the correlation coefficient or the coefficient of determination, R squared, the y-intercept, the slope, and you want to have an analysis of the deviation from the actual data points. So you want to, we do residual sum of squares, which is a comparison of the calculated y against the known y, and that's how we report that. For low range limits, this would be the LOD and the LOQ. There's which is also the QL, quantitation limit, and detection limit. There's a couple of ways that you can determine those. One is by visual evaluation, and visual evaluation isn't suitable for HPLC or GC. Visual evaluation is for things like turbidity or maybe TLC, things where the endpoint, the assessment is visual. We typically use signal to noise, so we'll analyze 6 replicate spike samples at the LOQ, and the signal to noise needs to be greater than 10. And if it's greater than 10 and we have acceptable accuracy and precision, then that's where we establish the LOQ. LOQ can be higher than 10, a signal to noise of 10. I would recommend, if you're using this method, to try to try to target 12 or maybe 15, just to give yourself a little bit of wiggle room for changes in lamp intensity and things like that. Same with the LOD, it's a signal to noise of 3. We do that with a single spike sample or standard at the LOD. If the sample or the API or the drug product has significant inherent impurities, it's really hard to assess LOD in the actual API. So you might have to do a matrix spike or just look at a standard. You can also look at the standard deviation of a blank if you have some inherent or some interference. Standard deviation of the y-intercepts. And then over on the right, there's the standard deviation of linear response and the slope. There's a calculation you can do where the detection limit is 3.3 times the standard deviation of the response divided by the slope. And the LOQ is 10 times the standard deviation divided by the slope. So the LOQ must have adequate accuracy and precision. We're going to get into that in the accuracy and precision sections, or the accuracy sections really. You need to report what your LOQ is, and you need to report what your approach is. You also need to report your accuracy and your precision data. So if you're using the signal to noise approach, you're reporting your signal to noise for those N of 6, you're reporting the accuracy for those N of 6, and you're reporting the RSD for those 6. And then you also need to report the LOD and the approach. So for impurity methods, your LOQ, the limit of quantitation of your method, needs to be greater than or equal to the reporting threshold of your method. And what that means is that in ICHQ3A and B, it states, you go through, if you've watched the specifications or the impurities webinar, you go through a process to figure out, based on the maximum daily dose, what the reporting threshold of your method has to be. So you have to be able to quantify at or above, or you have to be able to quantify the reporting threshold. So that means your LOQ needs to be needs to encompass that reporting threshold. So I think it should be, your LOQ needs to be less than or equal to your reporting threshold, not greater than. Accuracy. So accuracy is a comparison of your measured results to your expected results across the range of your method. So you are going to look at, when you analyze something, how does it compare to what it was supposed to be? What is it compared to the theoretical concentration? The studies that you can do is comparison to a reference material. And comparing to a reference material is really just analyzing it against a standard. So in this case, this would be like what we would do for an API assay. We're not doing any spiking or anything like that. You can't spike a pure substance for the assay. The second one is spiking into a matrix. If you've got a drug product, you can spike onto the matrix across the range of the method. You can also do the orthogonal procedure comparison. And that would be a previously validated method. If you're validating for the first time, you might very likely not have that available. When you're spiking, sometimes for drug product, like one time we had a product that had a really, really high drug load, and we couldn't spike at that concentration onto the sample. The spike volumes would have been crazy. The constant spike concentration would have been crazy. So what we ended up doing there is, it was a soft gel. We had placebo soft gel that we cut open, and then we actually just weighed API onto it. So we were weighing, 50, 100 milligrams onto the soft gels. For accuracy, you want to do 3 replicates across 3 levels. So that would be low, and then we got low, mid, high, but it's low, it's the target concentration, and high. So if we are 70, 100, 130, then we are spiking at 3 replicates at 70, 3 at 100, 3 at 130. And we're reporting the percent recovery. So that is the measured to the theoretical times 100. And then there's also a requirement that we report the confidence interval. And you need to make an assessment of that confidence interval against the intended use, against your specifications. If you look at the confidence interval for these accuracy sets, and the confidence wider or uncomfortably close to the range of your specification, you need to look at whether or not that method is suitable for its intended use. And then on to precision. So precision, it should be using authentic homogeneous samples. I like to look at precision is, if we execute the method as it's written, can we get consistent results? Can I get consistent results? Can another analyst on a different method on a different day get the same results I did? And that's intermediate precision. Can another lab, reproducibility, get the same results that I got on different instrumentation, different analysts, all of that kind of thing, on a normal sample? Now, if you're testing for impurities, it's possible that you've got the cleanest API and there's no impurities in there. So you're going to have to spike some stuff. You're going to have to spike impurities onto your onto your product. You want to make sure, though, if you're spiking with the API at the nominal concentration, so you can make sure that it's as consistent, or you're spiking on actual product, not just matrix. So repeatability looks very similar to accuracy. 9 determinations, 3 replicates across 3 levels. So it's very possible that you can perform precision and accuracy using the same samples, especially if you're really lucky and you have maybe in-house formulations, in-house product development that can make you drug product at 70, 100, and 130% of the target concentration. Then you're really lucky that you can just test your drug product as formulated for your accuracy, and you're not having to do spikes. But typically the way we do it is we do accuracy, 3 replicates, 3 levels, whether it's spiked or nominal. And then we do an N of 6 at 100%. And that 100% is, for assay, a normal sample. It's a normal lot. So we would combine those 2 studies in the protocol and say we're going to do an N of 3 at 70, an N of 6 at 100, and an N of 3 at 130 for assay. So reporting, again, you can combine accuracy and precision. I typically would report accuracy, and then I would come in and have a separate table that does my method precision, so my N of 6 for the repeatability, and then the intermediate precision in the same table, so we can make a direct comparison. All of the data is there together. So I re-report my 100% in the method validation report. But you can combine those. And then when we're looking at method precision, which is the repeatability, and intermediate precision, I like to look at the pooled standard deviation. I like to do that for impurities specifically, or looking at percent difference. So, and the reason for this, if we're looking at impurities and the impurity level is really low, let's say it's maybe 0.1%, 10% of 0.1% is 0.11, right? Which isn't really, it's not outside of the range of probability when you're looking at 2 different analysts. So you could, at low concentrations, have an inflated, it might look like you're failing intermediate precision when really it's still appropriate for its intended use. And that's where pooled standard deviation comes in. All right, and then robustness. Robustness, I think I said this already, it's the reliability of a method in response to deliberate variations. So we see this as slight changes in chromatographic conditions like injection volume or flow rate. We also can see it in extraction time or temperatures, things like that. Making sure that you're not on the knife's edge with anything. You're not, if you have, if there's something going on with your injector and your injection volume's a little bit low, you're not going to start failing LOQ. You're not going to lose that sensitivity. And then it's also stability of your samples and standards and reagents. The ICH states that you need to have stability of your samples to 24 hours for any method. If you can't get that, then you need to provide some sort of justification of how you're going to control it, whether it is a limit on the sample analysis time or perhaps specific special storage for your samples. All right, we're going to run through now in the last 10 minutes the different types of methods and how we approach method validation. And I'll try to give you some specific examples of when it gets kind of complicated. So for identification methods, specificity only, we're looking at the blank. If it's a product, looking at the matrix. And then other similar analytes to make sure that we wouldn't accidentally give it a positive ID when it's a different, maybe that's specific to like chiral APIs. And then as appropriate, we would look at the impurities and make sure that we have specificity of impurities. For a limit method, so this is a qualitative method for an impurity, as an example, We would be looking at specificity, blank matrix, structurally similar materials, and then we need to do a positive and negative confirmation. So we want to make sure that something that is above the limit, it fails, and something that's below the limit passes. So you're going to run two sets of samples. I've seen it as an N of 1 of each of them. I've seen it as an N of 3 of each of them. And then you want to establish your LOD. And this is where I mentioned earlier that there was a difference between the USP and the ICH. One of them requires LOD determination for limit methods. The other one doesn't. I say determine your LOD. I think that is important. If you're saying it's non-detect, you better know at what level you can detect. All right, the simplest in theory API assay, or the hardest, depending on the year. API assay, we're looking at specificity of your blank and your impurities. And your specificity of your impurities only needs to be to the API. It doesn't need to be to each other. They can collude, doesn't matter. Linearity, 80 to 120% of the target concentration, specification range. If your spec range is really, really tight, yeah, you just need to go outside of that. Accuracy and method precision, we lump these two together. So we typically do an N of 3 at 80, sometimes it's 90% and 120%, with N of 6 at 100%. And this is where our method precision and our accuracy come together. There is a statement in the ICH that says that accuracy can be implied by the establishment of precision and linearity across the range of the method. So the way that we apply this, you can't spike a pure substance. We can't spike the API onto the API. So we manage this by reducing the weight and increasing the weight. So if our target weight is 100 milligrams, we are weighing 80 and 120 milligrams for those lower and higher levels. And then intermediate precision is an N of 6 of the same lot, and we're doing that comparison of the average for a percent difference, or we're doing pooled RSD between the two of them. Next up is drug product assay, very similar to drug substance assay. This also is any other major analyte, so preservatives, antioxidants, critical excipients. We look at specificity, blank, we're adding matrix into this. We want to make sure that there are no matrix peaks that interfere with our ability to detect any of the analytes. And then the impurities specific to the API. And they don't have to be specific to each other. Linearity, 70 to 130 of the target concentration. Typically your specification for drug product is 90 to 110% of the label claim. So 70 to 130 gives you quite outside of that. And then accuracy, we're spiking, or again, if you're really lucky and you have a formulator that can make you product at 70 and 130, you're just analyzing an N of 3 at each of those concentrations. Then we do a separate, if we're spiking, we're doing a separate method precision, again, on an authentic sample. I always want to look at method precision and intermediate precision as the routine analysis of the method. So wherever possible, we're not spiking anything, we're just running the sample that we have. So N of 6 for method precision, somebody else runs it on a different day, maybe a different instrument, maybe a different lab, and you compare, again, pooled standard deviation or percent difference. This font's really small in this, I apologize. API impurities. We're looking at specificity of a blank, impurities to the API and to each other. And when we're talking about impurities in relation to method validation, it doesn't matter if it's a process impurity or if it's a degradant. We're validating for analytes. We're validating impurities at low levels. So it really, you can look at it not necessarily as an impurity method, but as a low-level analyte method, a trace level method, right? Doesn't matter if it's a degradant, doesn't matter if it is a process impurity. We're just looking at specificity. Can we separate everything out? Can we see everything? Our linearity, the range is the LOQ of the method, which should be lower than the reporting limit, to 100 greater than or equal to 120% of the specification for each one of those impurities. If they're commercially available, you're going to run linearity for them. If they're not commercially available, you can't run linearity for them. You also want to run your API or your analyte that is used to calculate unknown impurities. So if you're running a standard that you're calculating against and it's your API, then that also needs to have linearity. And you're going to use the linearity of your API and the linearity of those impurities. You're going to compare the slopes of those to determine your relative response factor if you need to. And the relative response factor, if you're not analyzing against the analyte itself, the impurity itself, you're analyzing against the API, there might be differences in responses. So we need to establish what the relative response factor is between the two of them. There is a statement in the ICH that says, I think, I don't remember exactly, but I think it is, if the relative response factor is between 0.8 and 1.2, so there's a difference of less than 20%, you don't have to apply a relative response factor. If it's outside of that, then you need to apply a relative response factor. You can apply a relative response factor, you just don't have to if it's within that plus or minus 20%. So accuracy is, for APIs, is us spiking impurities into the API. Sometimes you have oftentimes you have an inherent amount of the impurities that can make this challenging. So you're really not assessing the LOQ of the method because you're spiking onto something that maybe is already at 0.1%, right? So if necessary, we have to subtract the inherent amounts. This is an example of where there's no perfect way, unless you can get your API more purified. There's no perfect way to perform impurity validation on an API that has inherent impurities. So you have to assess what you've got and come up with a plan of what's the best way to approach this. Sometimes it means that you're doing two sets of something. So as an example, if there are some impurities that are present and I do accuracy of my LOQ, I'm still showing that I can see that difference, but I might do also some LOQ standards, replicate LOQ standards, to show that I can get signal to noise for those impurities when they're by themselves, right? So you're going to do the LOQ n equals 6. We always do LOQ at n equals 6. Mid and high, we do n equals 3. The high, again, is greater than or equal to 120, whatever the range is that you've determined. The mid, there's in one of the guidances says around the specification limit. If your range is quite wide or your specification limit is close to the 120%, I would do a midpoint more than the spec there, but again, you need to assess it. We don't want to run really low level and then two really high levels, right? So then we do method and intermediate precision. Again, authentic sample. If there's not enough impurities in it, spike it. It doesn't matter for method and intermediate precision what the concentration of those impurities are. Ideally, they are less than the specification, certainly within the range of the method, but it doesn't matter that you necessarily have all of them for method precision. You need to have some of them, and you want to look at not just the individuals, but you also want to look at the total impurities. So you want to calculate total impurities, and then we report everything, but we only assess RSD, and we only assess method precision to intermediate precision on any impurity that is consistently above the LOQ of the method. And with this, it is the I would do pooled RSD again for this because it is a low level. And then we need to do the limits. It is not required for you to do an LOD, but LOD is very useful for reporting. If you're reporting less than LOQ is the range between your LOD and your LOQ, and non-detect is below your LOD, you need to establish that LOD, but there's no requirement to establish it. But if that's how you want to report, you need to have that LOD established. And again, that's signal to noise greater than 3, using a standard, typically for each analyte, not typically, for each analyte. Again, you can spike that into your sample, but if you have inherent amounts of the impurities, you're just going to have to do a standard at the LOD. And the same goes for the LOQ. For the LOQ, again, N of 6, spike samples, sometimes it has to be a standard, RSD, signal to noise, and recovery. And the last one is the drug product impurities. Very similar to the drug substance, except that in some ways it's a little bit easier because we can just use the matrix to spike. So we have the blank for specificity, blank, matrix. Impurities need to be specific to the API and to each other. I want to restate to make sure that that I said it really clearly, that when you're looking at impurity analysis, it is critical that you are looking at the impurities with the target amount of the API in there. The reason for that is because you could have impurities that elute close to the API. And we have seen method validations where they have spiked into a matrix with and the LOQ would have been consumed. It would have been overwhelmed by that large API peak, because typically in our related compounds methods, our impurity methods, our API is at a really high concentration, so we can see those low levels. So it's really important that you have your API in there at your target concentration so you can demonstrate the specificity of those impurities with the API at its concentration. Linearity, again, LOQ to greater than or equal to 120% of the spec, each commercially available impurity. You can, if you have the same method, chromatography method, utilize your linearity from your API method. All of that is the same as what we already talked about. Accuracy, spiked impurities into the matrix with the API at the target concentration. So typically it would be spiking impurities onto your nominal formulation. LOQ n equals 6, mid and high n equals 3, and you subtract nominal impurity concentration. One thing that we do when we do accuracy studies is we run an unspiked. Sometimes we'll run like triplicate unspiked, and then we'll also run our spikes without any sample. And that just helps if something happens, maybe we had an error in a preparation, we're not attributing poor recovery to a method failure. Maybe we had an error in preparation of our spike. So we tend to do that. Method and intermediate precision, where possible, using an authentic sample. If you need to spike, I recommend preparing one single spiking solution and having both method and intermediate precision run using that same spike solution. When you're designing your method validation, you really want to make sure that what you are measuring is the execution of the method and not the analyst's ability to make a spiking solution. So if the method precision and the intermediate precision analysts are making separate spiking solutions, it's possible that there's a difference between the two of them, and you're going to attribute a difference to a failure in intermediate precision when in fact it is a failure in their ability to pipette small quantities. Reporting individuals that are consistently above the LOQ and the total. We don't assess. We did for a while, but we don't assess individual unknowns, so highest unknown anymore, because we would have situations where we would have two unknowns that were of similar concentration. It popped back and forth between the two of them and created a false sense of variability, right? Or the intermediate precision and the method precision, it was two different impurities. One was at 0.11, one was at 0.2 or something like that. It wasn't a good assessment of the capability of the method. So we went to total and just knowns for that. Again, comparing method precision and intermediate precision, in this case, pooled RSD. Same thing about the limits, the LOD, you can spike into the matrix maybe without the API in this case, or just running the spike sample or spike standard. And then LOQ, n equals 6 of spike samples. We need the signal to noise, RSD, and recovery at the LOQ. And that is it. I'll check and see if there's any questions that have come in, and we'll get back to you on those. I hope that you enjoyed this. If you couldn't tell, I do absolutely love method validation. I love crafting. I think that this is an opportunity for scientists to be really creative in crafting how they're going to demonstrate method suitability. So next up is IND, NDA, DMF. We're talking with Steve Pondell. He's going to tell us all about what is required for an IND and a DMF, and how you get from an IND to an NDA, as well as what analytical data you have to generate for those regulatory filings. Thank you so much for joining us.